Hi, everyone. Welcome to the Tuesday seminars on the cerebellum. Today we have as our guest Maxime Beau, who studied medicine and neuroscience at the University Paris Descartes, University Pierre Marie Curie, and Nomal Superior in Paris, where he worked with the laboratory of Boris Barber. After he received his MD, he went to University College London to study with Michael Heuser, receiving a PhD, co-advised with Beverly Clark and Dimitar Kostadinov, who we heard from last week. During his PhD, Maxime co-developed an important deep learning classifier to predict cerebellar cell types from their electrophysiological signatures on high-density silicon probes like neuropixels. He led a collaboration that involved laboratories of Dimitar, Dana Cohen, Kurt Hall, Steve Lisberger, and Javier Medina. At University College London, Maxime also studied monosynaptic information transmission from the Purkinje cells to the nucleus, where he's going to describe that work in a later publication. Maxime is going to come to the United States in a few months to study at Princeton University in the laboratory of Carlos Brody. Good to have you, Maxime.
Yeah, thank you for the introduction, Reza. Hello, Reza, and hello, everybody. So I'm very excited to present to you indeed two projects for my PhD, which I did at University College London in the labs of Michael Heuser and Beverly Clark, but also collaborating with the labs of Steve Lisberger, Javier Medina, Kurt Hall, Dana Cohen, and Dimitar Kostadinov, who did give his seminar last week. But before starting, I just wanted to thank you for these seminars. I think they're really awesome. They're a great opportunity to learn about the field, and for young trainees like me, they're really an invaluable resource because they sing on YouTube forever, and I really enjoy watching them. So thank you for doing this. And I also wanted to share my empathy about the current situation in the US for academics and scientists. I just hope that you're all okay out there, and because it seems pretty hard from a distance. Indeed, today I will tell you about two projects. I will tell you about monosynaptic information transmission from Purkinje cells to their targets in the cerebellar nuclei. But I will also, but first I will share with you our study, which was published in Cell last month, on how we devised a strategy to classify most of the major cerebellum cell types. Well, sorry, my cursor is bugging. Cell types. Sorry, can you see my laser? Oops. My computer is it's in the load from Zoom. Okay, I don't think I'll be able to use my laser too much, but I'll do my best. So we published a study last week on the classification of most of the major cerebellum cell types from the cerebellum cortex from silicon probe recordings. Okay, seems to have it covered now. So people attending this seminar probably know why the cerebellum is such an interesting circuit to study. It has a beautiful crystalline architecture and has only a few distinct cell types, as described by Cajal over a century ago. But despite this simplicity, fundamental questions remain about the specific computational role of each of its cell types. This is true for all cell types, but in particular, it is unclear whether Purkinje cells, in blue here, affect the output of the cerebellum according to a fast rate code relying on inhibition, or a temporal code relying on entrainment of its targets in the nuclei. So in the first part of my talk, I will tell you about the strategy we developed to identify cerebellum cell types on silicon probe recordings. And in the second part of my talk, I will share with you our findings on how Purkinje cells monosynaptically influence their targets in the cerebellum nuclei during behavior. But the thing is that we have been able to record from these neurons for decades. Here is the first extracellular recording of a Purkinje cells, simple and complex spikes by Granit and Phillips nearly 70 years ago. However, it has proved difficult to reliably map these recordings back onto these primary cell types, especially simultaneously and engaged in controlling behavior, to directly test this hypothesis. Several groups have tried in the past, but with mixed success. And now with modern high-density silicon probes, such as neuropixels, we can record more cells than ever before at a higher resolution than ever before. But we're still faced with the same problem. So we decided to take advantage of developments in reporting technology, of course, also optogenetics and machine learning to try again. This was the end goal, figuring out how to identify cerebellum cell types from their electrical properties on a probe. And spoiler alert, we cracked this problem for most major cell types of the cerebellum cortex. Our algorithm can successfully predict the identity of Purkinje cell simple spikes, complex spikes, molecularly interneurons, Golgi cells, and mossy fibers. But to reach that point, we had to address several issues that we solved along the way. Our first challenge was to generate a high-quality ground truth database of cerebellum neurons recorded with... Sorry? No, all good. And after having generated this ground truth database, we needed to then establish how to best exploit it to teach a machine learning model how to predict cell types from the extracellular properties before finally devising a strategy to validate our classifier on independent data sets from a different lab and also from a different species. So diving right in, the core of our ground truth collection is a technique called optotagging. I don't know if my screen is updating for you. Yeah, I think I just have too many slides. My MacBook is struggling to keep the pace, but I'm just gonna go, yeah, I'm gonna wait every now and then. So optotagging, oops. Yes. So we use different mouse lines to express specifically channel rhodopsin in Golgi cells, mossy fibers, molecularly interneurons, and Purkinje cells. But we needed to guarantee that the responses we saw to the optical stimuli was direct due to the channel rhodopsin expression and not transsynaptic. So we also washed on synaptic blockers and used the responses to the optostimuli during the synaptic block exclusively to tag neurons. Starting with more than 3,000 manually curated units recorded with neuropixels, we end up with 202 neurons distributed across these cell classes. And without getting into the weeds of why the attrition rate of units is so high, just remember this. It's freezing again. So remember that optotagging is a challenging technique when paired with silicon probes, and you need to do many recordings in order to... Yes, sure. I'm just going to turn off my video to see if it works, because I'm told that you guys are struggling to hear me too. Yeah, I'm just gonna stop screen sharing for a second, turn off my video, and come back to you. Okay, this is what it's better now. Here we are. Oops, stop sharing, there's the wrong screen. Okay. All right. So optotagging is a challenging technique when paired with silicon probe recordings, and you just need to do a lot of recordings to make that work and end up with a sensibly sized database. And finally, equipped with this highly curated ground truth database, we then proceeded to devise a model which would allow us to map the extracellular properties of neurons onto their identity. So this is the starting point, our ground truth database, which you can see here on the screen. On the top row, you can see the average waveforms of neurons for each cell type. Purkinje cells, simple and complex spikes, molecularly interneurons, Golgi cells, and mossy fibers. And on the bottom row, you can see their autocorrelograms, which summarize their spiking dynamics. Importantly, we decided to stratify the autocorrelograms based on the instantaneous firing rate of neurons. David Hertzfeld, who is a co-first author on this study and might be here on the call, came up with so-called three-dimensional autocorrelograms. They allowed us to unpack the variance of a neuron's firing rate, for instance, due to behavior, typically, to get a picture that consistently reveals the underlying dynamics of each cell. So you can see that at first glance, there seems to be consistency within class and differences between classes. From that point, the classical approach would have been to manually design a small set of statistics to summarize all of this information. For instance, the waveform width or the mean firing rate of each neuron to try to predict their identity. But in ML jargon, this classical approach is called manual feature engineering. It allows you to compress the raw data into few statistics that you can then use to predict cell classes with a simple supervised model of any type, really. However, manually cherry-picking features has a few issues. It's hard work, so the approach does not generalize well across datasets or brain regions. You have to redo it all every time. It is also prone to human bias, and it probably leaves information behind, as simple features never capture the full picture of the raw data. Instead, we decided to harness the latest developments in deep learning to predict cell type identity directly from the raw waveforms and autocorrelograms. A model that would learn how to compress this high-dimensional representation for us, removing the need to choose statistics ourselves. For that purpose, one could build a large model that maps from raw data to cell classes directly. But the issue with large models with many parameters is that they're very data hungry. As I told you earlier, optotagging is very difficult. And so we needed to find a way to work with our highly curated, therefore small, ground truth datasets. So we converged on the best of both worlds. First, compress the data, like with manual feature engineering, but in a data-driven manner with an unsupervised deep neural network that hopefully only leaves noise behind as opposed to information. And then a simple supervised model as a second half to predict cell classes identity from this compressed representation of raw waveforms and autocorrelograms that does not incur the risk of overfitting in a low data regime. So the compressor networks are variational autoencoders trained on thousands of enabled non-optotag neurons, which are readily available to reconstruct waveforms and 3D autocorrelograms passed through small bottlenecks. One network to compress waveform information and one network to compress correlogram information. And you can see that the reconstructions are denoised here, which reflects what I said earlier. Data-driven compression, when well-tuned, leaves noise behind, not information. So you can then chop these compressor-decompressor networks to only keep the compressor top halves and stitch them with a multi-layer perceptron, ending up with a full deep neural network predicting cell types from extracellular raw data directly. We then proceeded to test this model using leave-one-out cross-validation. We trained the model on all the ground truth neurons but one and predicted this one neuron cell type to build their confusion matrix, which you can see here. On the diagonal, chance level is 20% and the perfect score is 100%. So we're doing pretty well. But you can notice not as well for every cell type, right? In particular, there's confusion between molecularly oriented neurons and Golgi cells. And we figured that this confusion might come from two sources. It might either stem from ambiguous looking neurons, somewhere between our Golgi and molecularly oriented neurons, or it can reflect high confidence errors, real mistakes that the network makes. So if we could find a way to monitor how confident the model is for each of its predictions for each neuron, we could discard ambiguous looking neurons and reassess the classifier's performance only with neurons it feels very certain about. So to do just that, we then trained a collection of models on different subsets of the data and also initialized with different initial condition to build an assembly of voters for each neuron. So instead of spitting out a single prediction, our ensemble model now returns a distribution of predictions, which is way more reliable than a single prediction. Finally, you can take the ratio between the best prediction and the next best prediction, and you end up with a confidence index paired with each neuron. We can then use this index to threshold out the low confidence neurons. We will only keep neurons that this crowd of classifiers feels at least twice as confident about as the next best prediction. We're going to use a threshold of 2. So coming back to our original confusion matrix, you can replot the cross-validated confusion matrix with only those high confidence predictions and witness that the performance has increased across all cell types. What this means is that the high confidence neurons are also the correct neurons, which isn't necessary by design, right? As I said earlier. And like everything, this comes at a cost, unfortunately. On the right-hand side, you can see a new column, which shows the percentage of ground truth neurons, which had a confident prediction and therefore landed above the confidence threshold. So we lost quite a few molecularly oriented neurons and Golgi cells, as you can see here. But we're still closer to 100 everywhere on the diagonal, including molecularly into neurons and Golgi cells. Their confusion has greatly decreased, but not completely. Fortunately, we all know here that Golgi cells and molecularly into neurons belong to different layers of the cerebellum cortex, right? So we reasoned that if we added a third source of information, a third input, the anatomical location of each neuron in the cerebellum cortex, our problem would be fully solved. And so we proceeded to do just that using Phylum, a tool developed in Javier Medina's lab. And the confusion between Golgi cells and MLIs completely fades out in this case. We get an almost perfect score. So here is our final classifier. And if you want to hear about more details about it, I invite you to read the paper. But the critical next step was to test this classifier to make sure it does not overfit to our ground truth database. So we went on to test it on a data set from a different lab, from mice, but also from a different species. And to our relief, the classifier's predictions also matched human labels from the mouse datasets and from different lab, which means that, first of all, our ground truth dataset looks similar to those independent human labels, but also that a classifier learned real patterns from the ground truth datasets without overfitting to noise. And to our great surprise, we're glad to see that there was also a great match between the confident predictions from the classifier and human expert labels on monkey data from the Liesberger lab. Note that these expert labels were made by physiologists who were completely blind to the mouse ground truth data. The ground truth data wasn't even recorded at the time. And if you want to hear more about the monkey data, I invite you to read a couple of preprints that David and Steve put online recently, which will tell you way more about this part of the study. And finally, I really need to mention to you the most important of our tricks, which is collaboration. So this project really was teamwork. We're scientists from 5 different labs were involved in this project. And yeah, I just wanted to say that it's been a great learning opportunity to interact with everyone on this team during my PhD. It's really been great. And of course, I would like in particular to thank my mentors, Michael Heusser, Beverly Clark, and also Dimitar Kostadinov, who was a postdoc in the lab across my PhD years. To conclude on the first part of my talk, by pairing high-density electrophysiology with optotagging, we have established a high-quality ground truth database for major cell types of the cerebellum cortex and developed a deep learning classifier that predicts cell types from the raw extracellular properties directly. And importantly, this classifier was validated in datasets from a different lab and also different species, revealing striking similarities between cell types recorded in mice and in monkeys. And I just wanted to point out that all aspects of this approach are generalizable across brain region. If other groups were to use our optotagging tips and our model architecture on their own ground truth database, to build and then to use their own ground truth database, they could apply this to other brain regions. And all of our code is available as part of the package NeurPixels, the Python package that I wrote to analyze NeurPixels data, of course, with contributions from everybody on the team. But also, you can learn more about the classifier in the paper published last month, and on the website that we put together to allow you guys to study to go explore the database yourself. So this is the landing page. It's at www.c4database.com. And in here, you can do 3 things. You can, first of all, find the classifier documentation to run our classifier as it is on your own data. I just want to point out that you should read the data requirements section carefully. It's not just plug and play. You should make sure that your data is filtered properly before using our classifier in particular. But also you can find, but also you can find the data sets directly that you can download for yourself if you want to train a new flavor of this classifier or for other purposes. And of course, what's also very fun is that we put together a dataset exploration dashboard, allowing you to go explore some of the classical waveform and autocorrelogram features of all these various cell types. You can just hover over data points and you can see, for instance, the waveform of this one mossy fiber popping up when you're hovering over it to go explore the data used to train the classifier directly. So yeah, I invite you to go explore it and also to go check out NeuroPixels. If you have any issue running the classifier in any way, please do post an issue on NeuroPixels. We're trying to be reactive, and yeah, don't hesitate to bug us if you need to. So yeah, so if you have any questions about this part, I'm just going to rush a little bit because of these technical issues. So please hold that thought and ask them at the end, if that's all right. Okay, so now I would like to focus on one cerebellum cell type in particular, Purkinje cells, and how they monosynaptically influence the activity of their downstream targets in the cerebellar nuclei. The cerebellum acts as a feed-forward controller for the rest of the brain, broadcasting corrective signals from its output stage, the cerebellum nuclei. The nuclei receive a large portion of their inputs from the cerebellum cortex foliated around them, whose sole projection neurons are Purkinje cells in green here. Purkinje cells spontaneously fire at about 100 Hz and massively converge onto the nuclei, inhibiting them strongly with GABA. And in response to this constant barrage of inhibition, the cerebellar nuclei also discharge spontaneously at about the same rate. And despite the central function of this synapse, which really is the keystone of the cerebellum output pathway, we still do not know how Purkinje cells precisely influence nuclear firing. Classically, the Purkinje cells are GABAergic and therefore thought to inhibit the cerebellar nuclei. So here, say in the context of Ibelin conditioning, when Purkinje cell simple spikes decrease, nuclear firing increases. In this setting, the information carried at the cortical stage would be inverted down in the nuclei. According to a rate code, nuclear firing would be the mirror image of their Purkinje counterparts. But other evidence challenged this hypothesis. Armstrong and colleagues recorded functionally related Purkinje cells and nuclear neurons chronically from locomoting cats and showed that Purkinje cells and nuclear cells fired in the same phase of the step cycle back in the 80s. This correlation might be due to the shared MOSI and parallel fiber inputs to the cortex and the nuclei. Purkinje cells and nuclear cells could be positively correlated on average because of this shared volley of excitation. But the role of Purkinje cells would still be to sculpt down the activity of the nuclei, like in the inverter model. We're calling this model the common input or feed-forward motif model, depending on the day. But more recently, other groups devised an alternative hypothesis to explain why Purkinje cells and their targets might be positively correlated. Disinhibition from Purkinje cells might cause time-locked spikes down in the nuclei, effectively entraining them with small phase shifts. So actually, groups have shown that the nuclei can be entrained by synchronous disinhibition when stimulating Purkinje cells artificially with either electrical or optical stimulation. We will refer to the third model as the transmitter model. Information carried by Purkinje cells is transmitted to the nuclei according to a temporal code, but with a slight phase shift, with a slight delay. If you were to smooth both the Purkinje and nuclear rates, you would end up with the same picture, well, with the opposite picture as in the inverter case, and a similar picture to the common input model. So, but the truth is that these three models are convenient mental handles reflecting extremes, but in reality, they could all coexist in the same neuron pair, depending on the state of the circuits. To answer these questions, we need to record simultaneous pairs of monosynaptically connected Purkinje cells and nuclear cells under physiological conditions during behavior. And this is what this talk will be about. So first, I will discuss how we recorded candidate Purkinje nuclear cells simultaneously in locomoting mice before explaining a new general method that we developed to distinguish putative monosynaptic connections from spurious correlations on extracellular recordings, which is critical in situations where the neurons you are recording exhibit high levels of synchrony, of millisecond synchrony. And finally, I will share with you our results on the mode of information transmission between Purkinje cells and nuclear neurons during behavior. So we performed our recordings using NeuroPixels probes in mice experiencing locomotion and Pavlovian conditioning. We tracked locomotion using deep lab cuts on high-speed video recordings. We then reconstructed each NeuroPixels probe track using three-dimensional serial 2-photon sectioning, allowing us to register each electrode to the Allen-Brain atlas, as you can see here in 3 dimensions. But importantly, we performed the recordings using 2 probes at a time, sticking 2 NeuroPixels probes at a time to increase our chances to find connected corticonuclear pairs multiplicatively. So that was quite key to make this experiment work. And here is an example recording spanning 1 millimeter of tissue from the surface, not the surface, but the top is at the top in this case. In green, orange, and purple, you can see 3 units, which were spike sorted using kilosort 2 and manually curated with phi. The green and orange units are located in the cerebellum cortex, which we know from our reconstruction, and the purple unit is in the cerebellar nuclei. And upon inspection of these units' autocorrelograms and waveforms, you might recognize Purkinje cell and complex spikes from the first part of my talk. We can identify that these simple spikes and complex spike units both originate from the same neuron because simple spikes pause after each complex spike. In each recording, we then proceeded to compute all the cross-correlograms between all identified Purkinje cells in the cortex and all units located down in the nuclei, like these purple units. And in about 5% of cases, when we get lucky, we find that the spiking probability of some nuclear units decreases dramatically about 1 millisecond after each spike from the Purkinje cell up in the cortex, which are delays which are consistent with delays incurred by monosynaptic inhibition. So this is the correlogram between these 2 units, which are separated by 1 millimeter of tissue. Focusing on Purkinje cells up in the cerebellum cortex, we confirm the in vitro finding from other groups. Purkinje cells exhibit a high baseline level of synchrony, and these 4 Purkinje cells, you can see their autocorrelograms sorted by depth in green here. You can see that neighboring Purkinje cells are synchronous with each other, whereas distant Purkinje cells are not. If you zoom in, you notice that the cross-correlograms between these pairs of Purkinje cells have a stereotypical cat ears shaped double peak, which is consistent with ephaptic coupling, as reported in a 2018 study from the Rego lab. And in my hands, they really always look like this, Purkinje cells. The ephaptic coupling really seems to be very mainstream. We also find that pairwise Purkinje cell synchrony decreases with pairwise distance over a spatial scale that is consistent with the in vitro findings reported in this paper. So with this fact in mind, we can return to our candidate pairs of connected Purkinje nuclear neurons and look at them. So here you can see the same 4 Purkinje cells, this time colored differently, which are inhibiting 2 nuclear neurons with the cross-correlograms between Purkinje cell and nuclear cells in this gray area here on the off diagonal. Notice how the yellow and purple Purkinje cells are particularly synchronous, right there. So this means that a negative correlation between the pink Purkinje cell and the green nuclear cell here might very well be the result of the positive correlation between the yellow Purkinje cell and the pink Purkinje cell multiplied by negative correlation between the yellow Purkinje cell and the green nuclear cell here. So there's ephaptic coupling in between this pair, and we're wondering whether this connection is real or whether it is simply spurious correlation due to the multiplication between this and these cross-correlograms. So focusing on this pair of weak and strong inhibition, to measure the influence of Purkinje to Purkinje correlations on Purkinje to nuclei inhibition, we grouped Purkinje cell spikes depending on their coupling to the local Purkinje population. Here in blue, you can see spikes from the first Purkinje cell occurring far away from the spikes of the 2 other Purkinje cells, 2 and 3. In red, you can see spikes of the first Purkinje cell that are highly correlated or synchronous with the other Purkinje cells. Ranking all Purkinje spikes depending on the level of coupling to the local Purkinje population on the x-axis, we notice that the Purkinje to nuclear inhibition on the y-axis remains strong, even so spikes isolated from any other Purkinje cell in the area in the blue rectangle here. However, if we perform the same operation for the second Purkinje cell, for the second pair here, we see that the inhibition completely fades away when considering isolated spikes. You can see that a cross-correlogram completely flattens, revealing that the second Purkinje cell does not affect nuclear spiking probability alone without help from neighboring Purkinje cells, really. So we conclude that the top Purkinje cell is very likely connected to this nuclear neuron, whereas the second Purkinje cell is likely not. Summarizing all of our data across 6 mice, this conservative selection criterion excludes 107 candidate pairs which exhibit seemingly significant levels of inhibition when considering all spikes on the x-axis. That's significant 0.05 p-value here, but that completely fade away when considering isolated spikes only, which you can see on the y-axis. So in total, our dataset that we're going to discuss from now on comprise 57 corticonuclear pairs recorded across 6 mice that we call putative monosynaptic connections. And these 57 pairs are distributed across lobule 5, CRUS1, and simplex, while all the nuclear neurons of these pairs are located in the interpositus nucleus. Importantly, all the pairs exhibit an inhibition latency which is compatible with direct monosynaptic inhibition, which is on average, that's 1.2 milliseconds across our pairs. And all in all, we find that we can record Purkinje cells converging on the same nuclear neurons. We do record convergence. Yeah. We record up to 6 Purkinje cells with the same nuclear target. All right. So talking about Purkinje cells and the effect that they have on the nuclei, we find that you have this oscillatory pattern that you must have noticed before. We find that they entrain the nuclear targets, actually. We devised an analysis to understand whether this oscillatory behavior was due to the intrinsic biophysical properties of the neurons in the nuclei, natural relaxation after inhibitions, or to the GABAergic inhibition influence of the upstream Purkinje cell. So here, you can see 2 confirmed connections and observe that the oscillation frequency of the cross-correlogram and the mode of the Purkinje cell, the upstream Purkinje cell, seem to match. This period here matches with this peak here, and this period there seems to match with this peak here. So in this example, so let's take this Purkinje cell and split its spikes based on the subsequent interspike interval. So in this example, you can see that. So here you can see different groups of spikes. In light gray, you have a bucket of Purkinje cell spikes, which are followed by a fast interspike interval. And in dark gray, you can see a bucket of Purkinje cell spikes, which are followed by a long interspike interval. And when using either of these groups of Purkinje cell spikes to plot the cross-correlogram with the same nuclear neuron, you can see that there is a match between the interspike interval of the group of Purkinje cell spikes and the period between the first inhibition of the nuclei and the subsequent trough, which reveals that this oscillatory behavior is due to consecutive strong inhibitions of the same Purkinje cell and is unlikely to be due to intrinsic properties, intrinsic biophysical properties of the nuclear neuron. So, oops, sorry, we can then expand this analysis across all of our pairs, and you can see here the summary across every pair. On the x-axis, you have the interspike interval of the upstream Purkinje cell, so it would be the mean value of this light gray or dark gray bucket there. And on the y-axis, you can see the period of the oscillation in the Purkinje to nuclei cross-correlogram. You can see that they match really well. They all sit on the identity line across datasets. This analysis has only been done on 25 pairs, which are out of the 57 are those which display this oscillatory behavior. Importantly, the point is that Purkinje cells entrain nuclear neurons, and here is the distribution of the amplitude of the rebound. Well, the increase in firing probability rather after each inhibition, which can go up to 20% of the baseline of the cross-correlogram. And we can also confirm known findings from the literature that I mentioned earlier. Artificially stimulating Purkinje cells expressing channel rhodopsin can entrain connected nuclear neurons at their stimulation frequency, 40, 60, or 80 Hertz. So here, the power of the nuclei interspike interval distribution from periods where Purkinje cells are stimulated at 40, 60, or 80 Hertz. So here, it would be 40, 60, and 80, match in the nuclei the stimulation frequency in the Purkinje cells. We then measured whether the increase in firing probability by the nuclear entrainment was sufficient to overcome inhibition. And by comparing the red and the blue areas under the curve, we find that across all pairs, inhibition matches, which you can see in gray, or overcomes entrainment, which you can see in blue here. And last but not least, we went on to measure whether this was also the case during behavior. So we went on to measure the activity of connected Purkinje and nuclear cells during behavior, during locomotion, which we monitored using high-speed videography. And we also used deep lab cut, importantly, to track the precise position of the poles located ipsilaterally to our recording site up in the cerebellum. We applied a Hilbert transform to transform the row pole position from Cartesian to polar coordinates to recover the phase of each step cycle. But however, it's important to note that mice don't always locomote nicely. They often groom their face, they wander about on the wheel. So it's important to curate locomotion step cycles, which we did by applying agglomerative clustering to the extracted step cycles projected in a low-dimensional space, which allowed us to obtain groups of curated, consistent trials to study the tuning of neurons in the cerebellum, cortex, and nuclei. So here you can see a group of step cycles that did not look really great. And here you can see a group of step cycles that are very consistent and build our confidence in the alignment time that we use for our subsequent analysis. Here I'm simply overlaying this cluster of step cycles onto the same video again. Doing this, we can see here an example Purkinje cell and its downstream target in the nuclei aligned to the onset of locomotion swings. You can see that they are anti-correlated with each other, consistent with the inverter model. So here, 0 degrees, remember, is the onset of the swing. So you can see the behavior at the top. As the pixel's position goes down, the mouse is swinging its pole forward, and you can see that the Purkinje cells and nuclear cells are tuned to that to the locomotion behavior. Indeed, pairs of neurons are anti-correlated most of the time, but not all of them, importantly. A minority are pretty strongly co-tuned during locomotion, just like Armstrong and colleagues found in the locomoting cat. However, ranking these pairs of neurons according to the strength of Purkinje inhibition reveals that these co-tuned connected pairs exhibit a relatively weak inhibition. This suggests that these pairs might be correlated due to common input from Mossian parallel fibers to both the cortex and nuclei, whereas most pairs are anti-correlated and consistent with an inverter model. So I would like to note that some summary for my second part. I think we're almost on time despite everything. Sorry about that again, 44 minutes. We can record putative pairs of monosynaptically connected Purkinje nuclear neurons and developed a general method to distinguish putative monosynaptic connections from spurious correlations in recordings of synchronous neural populations. This would be useful in any system. And finally, we find that Purkinje cells can entrain their targets in the nuclei, but however, at rest and during behavior, Purkinje cell inhibition seems to prevail over entrainment. And I would like to note that we haven't talked at all about how Purkinje cell synchrony affects what we see. And also, I would like to report that the relationship between Purkinje cells and the targets in nuclei seems to be actually pretty different during leaking bouts. So there's still a lot of things to analyze. It's work in progress, and stay tuned for the end of the story. So again, I would like to acknowledge everyone who contributed to this study. Dimitar for Kostadinov for supervision, also Ricardo Rato, Hannah Stab, and Matthias Varadi, student that I supervise in the lab. Alex Fratzel and Rob Campbell for giving us early access to the 3D two-photon sectioning system that we've used to do our reconstructions. Anne Beverly and Michael and Dana for amazing discussions and supervision during my PhD. And of course, Caroline Reuter, our great lab manager who is keeping the lab afloat and who I really want to thank personally. And again, all the code for this project is also part of the NeuroPixels package, which you can just pip install if you want. So yeah, thank you very much.
Well, that was wonderful, Maxime. Really a fantastic talk. And despite our little bit of technical difficulties you had, you really did wonderful. So there's a question for you on the chat, but also if anybody has questions, just please go ahead, raise your hand, and I will call on you. I'll just read the chat for you. I don't know if you can see it. Maybe you want to look at it yourself. They're asking you a question about how to use the tool.
Yeah. So can this tool be applied to HD MEA recordings, referring to NeuroPixels package? Yeah. So any of the code that we use has been designed for NeuroPixels probes, but really should be compatible with any device that is compatible with a spike sorter that works with Phi, that is using the same data format. So you can use these spike sorters to spike sort any probe. And actually, the monkey recordings from David and I mean in Steve Lisberger's lab were not recorded with NeuroPixels probes. They were recorded with a different silicon probe. So you might encounter bugs along the way, and I invite you to post issues if you do. But in theory, it should work with any multi-electrode array. Yeah.
Maxime, thank you. Beautiful work and beautiful talk. I really enjoyed it. Especially the Purkinje cell and the cerebellar nucleus recordings are really amazing. So my first question is that, do you see a rebound after the suppression in the nucleus neurons? And do you do like you had your ZS scores on the y-axis, but I'm wondering how did you correct for the rate correlations between the two for the cross-correlograms?
So do I see a rebound after inhibitions in general? So I see what I showed on my slides, which is an increase in nuclear firing probability, which might be due either to a natural relaxation of the nuclear firing due to the channels making it fire spontaneously, or which might be active due, for instance, to calcium conductances. We don't know, and we're actually trying to answer this question. I'm wondering whether you were actually asking about complex spikes from Purkinje cells, or whether you were just talking about the simple spike that I showed on my slide.
I'm talking about the simplest spike, and actually, so you were showing that it seemed to be a fluctuation after and before in the cross-corelograms that looked to be matching the Purkinje cell dynamics. So yeah, what exactly. I'm asking is that, were you able to are you using jittering to remove the rate correlations between the two? Because this is the next spike that is causing the dynamics. So
yes, so there's an oscillation in the future and in the past because the Purkinje cell fires very regularly, and the past spike of the Purkinje cell also affects the cross-correlogram between the Purkinje cell and the nuclear neuron. So I mean, if I just oops.
So the rebound time is going to be dependent on the presynaptic neuron, not intrinsic to the nucleus neuron?
Yes.
Nucleus neuron?
Yes. So sorry, I think if you made this up again, it might not let me. Okay, can you see my slide again?
Yeah.
Great. Yes. So yeah, so indeed, so you can see that the deepest trough is the time of the inhibition from the Purkinje cell, but the trough in the future and in the past are both harmonics of the same trough due to how regular the Purkinje cell is. So you're going to have a very high probability of inhibition here from the Purkinje cell and also right there. And so you have a trough in the past and in the future. And what you can see on this plot is actually on the y-axis, the period of this cross-correlogram, and the x-axis, the periodicity of the Purkinje cell itself. And you see that all these points sit on the identity line. Actually, you can see triplets of points which are linked by a line. Each triplet corresponds to one Purkinje to nuclei pair that I have grouped into 3 buckets of interspike intervals, just like I did here, 5 in this case, but 3 across all neurons. And you see that as you take Purkinje spikes, which are followed by longer, like later and later second spike, you also delay the cross-correlogram period, where this gap becomes longer and longer, right? I don't know if that answers your question or whether you have.
Yeah, it does. Yeah, it does. So yeah, thank you.
Okay, thank you.
Maxime, I was really interested in the fact that you have multiple Purkinje cells converging on the same nucleus neuron. In one case, I think you had 6 of them. What
Yes.
can you tell us about the properties of those Purkinje cells during behavior?
Yes. So Purkinje cells are co-tuned to each other. Like Purkinje cells converging on the same target in the nuclei seem to be co-tuned with each other. I didn't manage to confirm this across all 5 mice who perform locomotion, but in these 2 animals, you can see on the top row the phase tuning on the x-axis, so 0 being the swing onset and pi being the stance onset. The tuning of each Purkinje cell that converge on the same nuclear neuron, which you can see on the bottom row here, with their exposition again being the tuning to the step cycle. And you see that data points, Purkinje cells that converge on the same targets, seem to be co-tuned with each other. Here you have this blue cluster in a mouse, and here you have this set of Purkinje cells converging on this nuclear cell, which is not co-tuned with this other Purkinje cell that converges on a different nuclear cell that is tuned to a different phase of the step cycle. So early results seem to show that Purkinje cells are co-tuned to each other, which is consistent with robust encoding of information to guarantee that their nuclear target is going to be, nicely tuned to locomotion. But these are early results, and I don't know if this will be confirmed across every animal.
Okay, Jorge, you have your hand up.
Yeah, hello. Do you hear me?
Yes, good to see you.
Hey, Maxime, good to see you. Also, thank you for the presentation. It was really cool. I have a bit of a question related precisely to that last conclusion of seeing that inhibition is what matters the most apparently for the Purkinje cell coding. And I wonder precisely that if they are all robustly synchronized or even modulating the same kind of information, if that's one of the reasons why you are seeing that, just because of the correlation of Purkinje cells is so high that the signal that it must be seen is only inhibition, and therefore related to the complex spikes. If we know that Purkinje cells are also having similar complex spikes, have you seen any maybe indication of complex spike pulsing or increases to the deep cerebral nuclei?
Yes, so I can start by talking about complex spikes. So here is the supplementary, showing the effect of complex spikes on nuclear neurons. And we can see several things. So I think somebody is speaking over us.
Yes, sorry, this is.
Oh, I see. This is in the background. Sorry. Here you can see a subset of connected Purkinje nuclear cells. In green, you can see the cross-correlogram between the simple spikes.
You can't see your slide, Maxime.
Oh, yeah. Oh, my God. I'm so sorry. Thank you, Reza. Oops. Yeah, is that the right screen? Yes. So in green are the correlograms I showed you earlier. Between simple spikes and the nuclear neuron, and in orange are the cross-correlograms between complex spikes and the nuclear neuron. And I find 2 interesting things. So the first is that you do not have in my hands a late rebound that would correspond to the pause in simple spiking. So we know that when a complex spike occurs, that there's a pause in simple spiking, but several papers, including from the Lang lab, have reported that Clemming fiber inputs, especially synchronous Clemming fiber input, should cause a rebound in the nuclei, but I don't seem to see that like too much, at least, or not the dramatic rebounds that one might expect. These are, again, early results. I need to quantify that more carefully. but this is what I can tell you. However, I see something interesting, which happens in two animals, which is this sharp excitation right after the onset of each complex spike. So after every complex spike, you have a sharp increase in nuclear spiking probability. But if you zoom on those cross-correlograms, so these are all the examples where I see that, you see that the latency of that increase in spiking probability is actually faster than the time it takes for inhibition to kick in. So in this case, it happens over 0.4 or 5 or 6 milliseconds, 0.4 in this case. And on the bottom, you can see a summary of the latency of complex spike to nuclear excitation compared to the time that the simple spikes of the same Purkinje cell take to have an effect on the nuclear neuron. So these nuclear neurons seem to fire more in a way that, like, before inhibition kicks in, maybe because of the collateral climbing fiber coming into the nuclei that would affect nuclear neurons before the Purkinje cell has time to generate a complex spike that goes down the axon. So this is an interesting finding, because it probably is consistent with the fact that a climbing fiber that innervates a given Purkinje cell also innervates its target in the nuclei, forming this closed loop motif that has been discussed in several papers, including in Shadmehr et al., and is a theory in the field. But I find this in only 2 animals, and it's quite anecdotal. So I will report this as a supplementary figure probably. What is really key is that we do not see these delayed rebounds in the nuclei followed by climbing fiber inputs, like complex spike coming from Purkinje cells going down in nuclei. Yeah. So yeah, that's kind of, I didn't have time to mention that during the talk.
That was wonderful, Maxime. Wonderful. Sasha, you have your hands up.
Great. So 2 questions, one a personal one and one a science one. Personal one first. Okay, you're going to Carlos Brody's lab. Does that mean you're leaving the cerebellar field, or that you're going to bring real thinking into the cortical field?
I'm going to expand my horizon, but I love the cerebellum and I don't intend to leave it. Actually, I'm going to record probably from the cerebellum of rats doing decision making alongside other brain regions. So yeah, I'm not fully leaving the cerebellum field.
Excellent, great. I think we're all happy about that. And you have a lot to teach Carlos about how to think about brain-wide dynamics. So looking forward to that. Okay, so you said at the beginning something that was really a gross overgeneralization, which is Purkinje cells fire at 100 hertz, and so do nucleus cells. So I take it from that your generalization comes from your own data set, and that you've been recording from a relatively homogeneous population of Purkinje cells. Probably they don't express aldolase C, and from a relatively homogeneous population of the largest dorsal interpositus neurons, yeah? And of course, you know that other cerebellar nucleus neurons, I didn't believe this at first, but now I do, can actually only fire at 10 hertz, even though they're glutamatergic projection neurons. And of course, Purkinje cells fire at rates that depend on where they are and who they are. So just little response from you on that. How do you think about how this particular model works as opposed to other models for other types?
Oops, sorry, I think, yeah, I can't have my video and share my slides at the same time, but I heard your question. I'm just going to turn off my video and share my slide up. Yeah. So yeah, I have not put my results in perspective with the average firing rate of the Purkinje cell that I've recorded. Indeed, it was a gross overgeneralization that I just used to introduce the problem. Indeed, you have subcategories of Purkinje cells depending on the zebrin expression in the cerebellum cortex. However, I will say that I do record from a distribution of lobules, lobule 5, CRUS1, and simplex, really not necessarily by design, because I've just tried to record from many, many places in order to compile my data set of 6 mice. Yeah, so I think it's like what is known is definitely that the nuclear rate affects the relationship between Purkinje cells and the nuclei. Work from Philippe Isope shows that these are probably the glutamatergic-like nuclear neurons, which fire at high rates that are likely to be entrained by Purkinje cells. I do not know whether the rate of the upstream Purkinje cell is going to affect their ability to entrain the nuclei. But as I showed you on this slide, whoops, let me come back to this result there. Yes, only 25 of my Purkinje nuclear pairs do exhibit this kind of oscillatory cross-correlograms. And you might need a very high firing and high regularity in the upstream Purkinje cells to elicit this behavior. So it's very probable that this result will depend on the rate of the upstream Purkinje cell, and we will definitely look into that.
Got it. Great. Public service announcement. Positive aldolase C or zebrin positive and negative Purkinje cells are in all of the lobules. So where within the lobule you're recording is much more important than which lobule you're recording from. So got it. And then I just wondered, with respect to the lick situation, there are really two types of Purkinje cells in the interpositus C region that have to do with licks. One is zebrin positive, the other one's zebrin negative. And so and they're really close to each other if you flat map the cerebellum. So just, I guess this is a stay tuned kind of question, but yeah, it's just like gorgeous work like everybody else says. So thank you.
Yeah, thank you. Yeah, I will definitely study the tuning of these pairs of cells in other behaviors because my mice are receiving rewards and they're licking to those rewards, and I see strong tuning to licking. I will try to also relate how tuned to licking, like how the licking tuning relates to the rate, therefore the probable belonging to aldolase C positive or negative microzones, because indeed they're in every microzone, in every lobule, sorry. Of the neurons. But yeah, thank you.
Wait, and just one last thing, because I hear you talk about rate, and I think that this is a common misconception because people record rate, but rate isn't what really distinguishes Purkinje cells the best, or any cell. It's the minimum inner spike interval, right? So if you can fire super fast if you can fire 2 or 3 spikes with very short inner spike intervals, your ability to time downstream neurons, which by the way, are similarly tuned, is much higher than if you're firing, if you can't fire at, so really look at minimum inner spike interval or firing capacity or something. That makes sense?
Yeah, so the sort of edge of the dynamic range of interspike intervals that the Purkinje cell can exhibit rather than the mean.
Exactly.
Okay, I'll look into that again. I'm a bit rusty on this precise literature. I was thinking about the Dizeu work mostly, but I might email you to ask you more details about this.
I'd be delighted. Thank you.
Thank you, Sasha.
Hi, Maxime. Beautiful talk. Thank you so much. I had a question about the cell classification model. So my understanding is, and correct me if I'm wrong, in the classification model, you mentioned that there was initially a problem with labeling between MLIs and Golgi cells.
Yeah.
And this was presumably due to off-target expression of GlyT2 Cre.
Yes.
And so you addressed this by incorporating anatomical information.
Correct.
But it's my understanding that Lugaro cells also express GlyT2. So how did you disambiguate between Lugaro cells, MLIs, and Golgi cells?
Yeah. So a limitation of our classifier is that we're only focusing on a fraction of the cell types in the cerebellar cortex. And it's like it is indeed like it is possible that out of distribution neurons that we do not know anything about, such as Lugaro cells or Purkinje cell, Purkinje layer interneuron or interneurons also might end up classified with high confidence as one of the cell types that we have. We cannot exclude that and we cannot verify that. So the answer is we do not know, but we're relying on how rare these neurons are compared to Golgi cells and molecularly interneurons to hope that the glycated positive cells that we found in the molecular layer are molecularly interneurons, and the glycated positive cells that we found in the granular layer are Golgi cells. But yeah, that's a limitation, and we did not do any form of sub-clustering of the optotagged ground truth dataset in order to include some or exclude some. We just kept everybody as is, apart from segregating them based on the anatomical location, of course, as you mentioned already.
But so if I can chime in here, we did actually exclude cells in the Purkinje layer from those. So
Yes.
they had to be so GlyT2 optoresponsive cells had to be very high in the molecular layer or very low in the granular cell layer, and so I think that we're actually excluding from the ground truth our things like potential Lugaro cells based on that anatomical designation. So we actually did think about this and account for it a little bit, but
yeah,
as Maxime says, it's probably not totally perfect.
Yeah, Lugaro cells are at the top of the granular layer, close to the Purkinje layer. Therefore,
that's right. That's right.
Excluded by our approach because we had a buffer zone. If the dataset looked ambiguous or seemed to be close to a Purkinje layer, we just excluded it. We just excluded those neurons. So this helpful Lugaro cell specific.
Yeah, as Maxime showed on that attrition slide, we were kind of ruthless about eliminating false positives.
Yeah.
I see. Thank you so much.
Any other questions? Well, Maxime, that was just wonderful. What a wonderful way to learn about this new tool and the website that you set up. I'm looking forward to the whole community using this and giving you feedback and improving it for everybody.
Yeah, let us know when things break, definitely, because they will.
All right. Thank you, everybody.
Thank you for the invite.